the results of the study, a discussion of the results, and my conclusions that I drew. First, the background. Wasted energy. In the United States alone, in 2012, an EPA did a study that 36 million tons of food waste and 33 million tons of yard trimmings were sent to landfills each year. Of that, only 4.8% was recycled of the food waste, and 57.7% of the yard trimmings were recycled, mainly into compost. And much like the rest of the society, colleges and universities produce these same types of waste on campus. The different types of organic waste that are produced, you have pre-consumer waste, which is waste that is before it reaches your plate. Things that are in the food preparation, spoiled food, things that can't be sent out. Post-consumer waste is all the leftovers that are on your plate, things that you didn't eat or that could not be consumed. And there's also landscaping waste, which consists of grass clippings, leaves, tree parts, prunings, replacements. On RMU's campus alone, we have 5 on-campus cafes and 1 at the Neville Island facility. We have, as I said, 6 cafes, 1 bistro at the Ice House, and 230-acre main campus. So you can imagine how much waste is produced, not only food, but also landscape waste. Anaerobic decomposition is a process that occurs naturally, and the word anaerobic just means without oxygen. So the bacteria are breaking down the products without oxygen. Biogas is a result of that. Specifically, methane can be produced as a result of the breakdown process. And the methane can be used as an alternative energy source. When these materials are sent to landfills, however, this biogas just escapes into the atmosphere. It can't be used for anything and actually can pollute. So anaerobic digester systems can be built that simulate the conditions for digestion, but that contain the whole process. The input materials are key for a digester. The more herbaceous the material, the more leafy or green the material, the more methane that can be produced from it, a higher biogas yield. Something like a lignin or lignocellulose content, that's more of the woody parts of plants and organic waste, that limits the yield of biogas. The ideal pH level in a digester is neutral, 7, and the entire process can take about 35 days from input to output. The residual material that's left after the composting process can actually be used as a fertilizer, as compost. So why an RMU digester? Well, just from the input from the campus dining facilities, the campus groundskeeping, and the surrounding community, we do live in a large metropolitan area. If we had some contribution from outside sources, such as food waste, yard waste, that could all be contributed to a campus digester that would not only produce an alternative energy source on campus, but the compost produced could also be shared with the community. And all of this waste could then be removed from local landfills. For my experiment, materials and methods, I constructed 3 small-scale digesters, and each digester was constructed from a 5-gallon plastic bucket with a lid, a plastic hose connected with some valves and some clamps to section parts of it off, and at the end of the hose was a 2.5-gallon collapsible water container to collect any of the gas that was produced during the process. And the entire system was sealed off to be airtight, so it would not allow gas to escape or gas to enter. The input materials that were chosen, each digester had a different input material. The first one was a representative sample from the RMU Cafe. It was mainly consisting of ends and pieces of watermelon, pineapple, some bits and pieces of beef, and also some mushrooms. Fruit was in the second one. It was apples and cantaloupes. And that was chosen because both of these products are highly fermentable and can produce a lot of gas. And third was a bucket with greens in it, which I chose celery and spinach to represent leafy greens and yard waste and the high herbaceous material. They also have a lot of cellulose in them, which again can produce a lot of biogas. All of the material was shredded using a blender and added to each bucket with equal parts water to fill about 2.5 gallons of the bucket. And then I had a sample of horse manure, which was 16 ounces, and I made a slurry with water and equally contributed that to each bucket. This provided a bacterial starter culture for the actual process to start working. Again, all the buckets were sealed and then were placed under a fume hood in one of the laboratories on campus. The ambient temperature was monitored with a thermometer, and the fluctuation of the temperature went from 5 degrees Celsius to 24 degrees Celsius, depending on the time of day and how cold it was outside. The average temperature was about 18 degrees. The material was allowed to digest for 14 days before I started to take samples, and after that I took samples every 7 days with one period where it was 14 days in between. Each sample consisted of 4 microliters of gas in a syringe, which was then taken to a mass spectrometer and gas chromatograph in one of the chemistry labs on campus. The results of the samples, again, 4 samples were taken, and each one of these lines represents a different, the top 3 lines represent samples, and the bottom one is just the ambient air that was around in the lab. And as you can see from the blue line is the cafe sample. It started out a little bit high, it rose, and then it dropped dramatically. The fruit just declined the entire process. The only one that had any progress, any increase in gas was the greens, and that was very minimal. As you can see, compared to air, there really was not a lot. So over the 30-day trial period, every time I took a sample of gas, I then evacuated the collapsible container to make sure that it was a new, fresh sample. No significant amount of methane was produced at all during the entire process. The collapsible containers themselves actually never inflated. And as I said, the fruit in the cafe samples decreased, and the greens is the only one that slightly increased. So what happened? I believe that there were 4 different possible problem sources. One being the temperature, 2, the location, 3, the vessels themselves, and 4, the input materials. The maximum temperature reached was 24 degrees Celsius, and it had fallen as low as 5. For mesophilic, that digestion, which is what I was aiming for, the range should be between 25 and 40 degrees Celsius, so it never even reached the bottom of that range. In an attempt to raise the temperature, I closed the vent hood, which again was letting in the outside air in its cold months, added a bottom heating mat, and also added some incandescent lamps, and none of these significantly raised the temperature. And other methods that we had thought of, including space heaters or other types of heating sources, just were not feasible for the space that we had provided. So in the future, experiments should employ reliable heating sources and a better way to moderate and to monitor the temperature. Another problem was the location. The digesters were all under the fume hood, which was vented to the outside air. This is the cause of the decreased temperature mainly. I mean, all the air was coming in from outside, and it was cold days, cold nights. So in the future, a more ideal location should be considered. Somewhere that's warmer, somewhere that has more space that would allow for better temperature control. And if possible, during the summer months, it would take care of both the safety concerns and the temperature concerns. The vessels themselves, the bucket system, they were completely sealed, which again posed a problem because there was no way to be able to monitor the pH or adjust the pH. So due to a high or low pH levels, it could have decreased or completely limited the biogas production. For the future, models that are more accessible to allow for monitoring and adjusting the pH should be considered. The input materials themselves, meat can actually lower pH. So again, I could not monitor what the pH level was, and that could have been a problem. So a better simulation of campus food waste could be done than what we had. Also, an addition of herbaceous landscape material would also help. And probably the most significant improvement that could be made is to get a mesophilic anaerobic inoculum from an existing system, the starter bacteria, to actually jumpstart the whole process. Excuse me. My conclusions, learned a lot of lessons from this study. The lack of biogas production was a result of probably more problems than I even have thought of. Excuse me. However, the possibility still exists that this is a feasible idea for RMU's campus. Future studies should be conducted to improve upon the research that I've done. And also another aspect that I did not even look into was the economic aspect. So another future study should study the economics of having a campus digester. Thank you for your time. Any questions?